All right, I'm actually really excited that Arnie put us all together because as you're going to see, we've been in part motivated by the extreme inadequacy of existing approaches to understand the repertoire and functionality of tumor-specific T-cell responses, which are absolutely central to the revolution in cancer immunotherapy, that we've started working on some of these new assays, which are generating some really deep, rich data sets, a lot of information, that we really now are at the point where we want to understand how those outputs ultimately relate to what is ultimately what matters to patients, which is clinical benefit. The assumption here, which I think is a totally reasonable assumption, is that the key drivers for a successful anti-tumor response are T cells. Just to give some background, because I realized actually last evening at dinner that in particular some of the folks from Microsoft and others aren't totally aware of what a transformative revolution for cancer treatment immunotherapy has been. It'd be interesting to get Jed's perspective on this as the other old guy in the room doing immunotherapy for like a quarter century. Actually, he's a lot younger than I am. I sort of personally had to go back to the antibiotic era to think of a time when a medical interventional technology has so rapidly, literally in a matter of a few years, transformed the way an obviously very important disease is treated and the outcome. I think the world, the research world, really only recognized this in 2013. Science's breakthrough of the year in 2013 was cancer immunotherapy. I went back. They almost never, almost all the science breakthroughs are a discovery. This was actually a field. And I love this quote of mine that somehow got into the Wall Street Journal. These days there are two kinds of pharma, the ones that are into immunotherapy up to their eyeballs and the ones that want to be. So this really is big. And just to give you a sense of, so, turn this off. Okay, I'll tell you what, I probably need to turn off the light. Okay. We hold our breath. Okay. Yeah. So up until probably roughly 7 years ago, if you had metastatic melanoma, you got chemotherapy, and this is kind of what your survival curve looked like. This is a Kaplan-Meier curve. So from time of therapy, time, survival. Then really the first immunotherapy that was widely available. IL-2 had its dent, but I think what really started changing the game were these antibody blockers of these immune checkpoints. Anti-CTLA-4 in 2010, the randomized trial. This is now going out 8 years. It's actually out as far as 10 years. Is this plateau of survival? So 22% of patients with metastatic melanoma you can use the term cure, but you can say they are living relatively normal lives with or without, some residual cancer. But we now expect that everybody's got to die, but they're not going to die of their cancer. The PD-1 pathway blockers, which in many ways have exploded the field based on their activity in many different cancer types. In second line, so this is basically after chemotherapy, look like they're plateauing out at about 35%, and it's still early for first line, so the first therapy that you get, and this is now standard of care. Patients with melanoma in general, the first therapy they will get is one of two anti-PD-1 drugs, either nivolumab or pembrolizumab. And that looks like it's going to plateau out at around 50%. So basically, we've gone from 5% to 50% plateau long-term survival in literally about 5 to 7 years. I mean, in the context of medical advancement, that is basically a heartbeat. Nobody ever thought that immunotherapy would work in lung cancer, but it does. And again, this is what you saw with chemotherapy, and this is now actually the long-term follow-up. It's, I think, 5 years. Looks like the plateau is out at about 16-17% survival, which compares with about 3% at 4 to 5 years.
Individuals or individual studies?
The dots are individuals. So the way these plots work is you start at 100% at the time of therapy, and if you die, the tick goes down. These particular dots represent individuals that are that many years out at the time.
Trying to get a sense of how many people involved.
Sure.
How does no treatment compare to chemo? Just out of curiosity.
How does what?
No treatment compared to chemo, out of curiosity. And chemo's pretty bad, it looks like.
In melanoma, it doesn't contribute much at all. But in lung cancer, chemo was the first intervention to improve survival. That doubled it.
I see. So no treatment, it looks the same as chemo in the melanoma?
Right. In lung cancer, no treatment would look like this. The key is that what chemotherapy does is the benefit is measured through median survival, which means the 50% point is moved out. But chemotherapy, when I think back to the number of adults with solid malignancies that I treated with chemotherapy that I would say are cured, I can count them on, it might take a second hand. So what chemotherapy virtually never does is give you this plateau. Now, there are a few patients, they're referred to as extreme responders. Everybody in the genomics world is interested in extreme responders. My own personal theory is that extreme responders to chemotherapy are actually really immunotherapeutic responses. They're responses in which you somehow ticked the patient's immune system over, and it's not that chemotherapy really killed all the tumor cells. It's really that you somehow broke that tolerance barrier, and it's the immune system from the get-go. But this is huge. It's terrific. But of course, that's still 80 to 85% of lung cancer patients that we haven't benefited, and on and on and on. So currently, checkpoint blockers are approved in 7 different cancers within, let's say, 2 years. there will probably be another 7 more approvals.
Are they used off-label also, or not really?
They are beginning to, and again,
But it's
Alex and
probably
Jed
hard
can
to get
speak
the
more
data
to
on
this.
that, right?
Where they are beginning to be used off-label is when it's clear that there's going to be an approval. So,
I see.
for example, Merkel cell cancer is one of the, for which there's a 55% response rate to anti-PD-1. Merkel cell cancer, when the New England Journal paper came out, that was about a year ago, there was just one approval. There will be Pembro, it's got to be any day now. He's just smiling. But I mean, I can tell you that our Merkel cell patients get Pembro, and the insurance companies actually are paying for it because it's clear that it's going to be approved. This kind of wild off-label usage is generally not applied, which is good. There are currently 5 checkpoint blockers that are on the market since the first. Actually, if you include IPI, I think 5. IPI, Pembro, Nivo, Atezo, and Merck Serono's is what's Avelamet. They're greater than, I think the number now is more like 1,500 immunotherapy trials, of which over 1,000 combination trials. It's insane. It's absolutely insane.
How many people are in each of these trials, on in average? Is that 10,000 patients? Is that 100,000 patients? A million patients?
Man,
Not a million.
it is actually becoming an issue of entering, accrual of patients just because there are so many trials, number one, but by the same token, it's still only about 4 or 5% of Americans with cancer actually go on a clinical trial, which is way too low. And now that a lot of these trials are moving immunotherapies to a much earlier stage, this whole patient education piece of what we need to do so that patients don't view clinical trials as their guinea pig, as depriving them of the best available therapy, as something you do as a last resort. One of the huge tsunamis coming forward is going to be neoadjuvant immunotherapy, so in patients that are surgically resectable but with a high relapse rate. So there's all kinds of things happening literally by the week. It's absolutely.
This data is owned by the drug companies, right?
Depends. A lot of it is. And I would say it's probably somewhere in the low to mid hundreds of thousands of patients right now.
Which is, again, it's huge.
That's a lot.
That's a lot.
Be a lot of data if you could actually get it good.
Oh, absolutely. So this is the pace of approvals, and you can see why this is a tsunami. So it started with IPI in 2011, but look, as you go along, it just gets faster and faster and faster. And this last year, roughly half of the approvals for cancer therapy, for cancer treatments were actually immunotherapies. I think it might have actually been over 50%. Now, just to make the point that this looks like a breakthrough, but all of these clinical successes in immunotherapy, and Mark and I were talking at dinner last night, he was saying, all of this great advance in cancer, in immunology, if you go into the clinic and ask, what are the immunologic analyses that doctors do, it's like white blood cell count. And so really this is a case where cancer, which again, cancer immunotherapy was just, you might as well have had leprosy if you were a cancer immunologist at an AACR meeting, 7 or 8 years ago. And now we're the coolest kid on the block. So, that's right. We are the living revenge of the nerds. And it's actually not true because we're now married to
So are
the
people
genomic.
in AI, by the way, it's the same. No, I'm serious.
All right, so we're a match made in
You're right,
heaven.
absolutely.
But I think it's important to make the point that these advances in cancer immunotherapy, which again look like there's just this sort of tsunami coming, really are built on probably 40 years of basic immunology and cancer immunology, elucidation of monoclonal antibodies, elucidation of what are tumor-associated antigens, now neoantigens, which comes from genomics, how T cells recognize antigen, and then it really was only starting in the 1990s that immunologists began to really view the regulation of immune responses as including breaks. So before that, we sort of looked at the immune response as an accelerator. Antigen came into the system, you activated the immune response. When you cleared the antigen, there was nothing more pushing the immune system, so it kind of petered out. And CTLA-4 was really the first example of a clear checkpoint. There have been other negative regulators, but this has obviously been a very recent set of advances, this elucidation of immune checkpoint. By the way, the cell replication folks hate us for co-opting the term checkpoint for immunology, but my feeling is live with it, guys.
What is your thinking about small molecule inhibition of checkpoint inhibitors? Is that a possibility or?
So I have always been very skeptical about the need for small molecule inhibitors of checkpoints that are either secreted molecules or cell membrane molecules, because antibodies seem to do that perfectly well. There are a lot of inhibitory intracellular signals. IDO is one of the really interesting ones. There are other very interesting metabolic inhibitors that are intracellular. So there's a whole world of intracellular checkpoints. Those do need drugs. So it's not like we don't need small molecules. I'm not personally so sure that a small molecule is going to beat an antibody for a conventional cell membrane, but that's just me. So this is not obviously all of the checkpoints, but to make the point that in the tumor microenvironment, this is a very three-dimensional thing. There are many cells involved. There are many molecules involved. So one of the areas in trying to figure out, there are now probably 50 molecular targets for immunomodulators that are being tested, mostly in combination with PD-1 pathway blockers. Again, over 1,000 combination trials. It's absolutely insane.
So this is explaining to us how the different types of possible checkpoint inhibitors, or
Can you walk us a little through this picture?
or tell us it's
So,
not necessary that we understand.
right, so I'm not going to walk, just because it'll take,
I mean, we want to see it for later. We want your slides for later. But
you can have all of these slides.
Yeah.
You can have all of these slides.
we'll write an email and ask you questions.
Right. The point of this slide is to say that within the tumor microenvironment, which is one of the areas where there is rich information about what is going on and what are the elements of the immune response that are engaged or that are being blocked in Mrs. Smith's versus Mr. Jones's tumor, is that there is a very complicated three-dimensional multicellular multi-molecular set of interactions. So one of the big pieces here is how do we transform the analysis of the biopsy, which into something and take advantage of that much richer amount of information that involves not only expression of molecules, but also the geography and geometry of the expression of those molecules, what cells they're being expressed on, what cells are next to other cells. So that's one big area which is not the topic of today, but one that, again, is very rich in terms of image analysis and computational biology. And this is where the astronomers and the pathologists are making music. So there are currently three major types of biomarkers that are being looked at very seriously and clearly have predictive value for response to anti-PD-1. First and foremost, expression of one of the two ligands for PD-1, PD-L1. There are now 4 FDA-approved PD-L1 immunohistochemistry tests. They all use different antibodies. They all use different, Merck's test looks at expression on tumor cells. Genentech's test looks at expression on non-tumor cells. It's a little bit of a Wild West. But I think, and some of these are used as companion diagnostics, some as complementary diagnostics, which is you can use it to help inform you, but it doesn't have to be used as a companion. I think the real breakthrough in appreciation for the value of this was the recent approval for pembrolizumab as first-line therapy in lung cancer in patients who, by Merck's companion PD-L1 diagnostic test, have greater than 50% positivity on their tumor cell. Is that, am I right in that number? It's greater than 50% positivity, right. So, but also now people are getting very interested in mutational burden, which clearly correlates, and CD8 T cell density. There's no formal FDA approval for mutational burden, but Foundation Medicine is now actually reading out calculated mutational load based on their, 300 or so gene panel, which clearly is meant to provide information to go into decisions about ordering, about whether to use immunotherapy sooner rather than later in the course of treatment of a...
Do you believe that there is something which is more predictive that you just haven't found that correlates with mutational burden? I mean, that there are
Yes,
particular...
hang on.
Okay.
Hang on.
Good.
And then there's CD8 T cell density, but there's more. There is T cell repertoire. So this is a wholly untapped field, and obviously the hypothesis is that mutations make good antigens, but there's the other side of the coin, which is the T cell repertoire. There's T cell function.
PD-L1 expression, is that, it happen all the time, half the time? In other words, the tumor cells have got to be expressing PD-L1 in order for that particular blockade.
Yeah, so it depends on the tumor type.
Okay, so it's correlated to tumor.
Yeah, some tumors it's a lot, some it's fewer. The other is the
Understanding of why?
right. It looks like the major mechanism for PD-L1 overexpression in tumors, and in some cases it's the tumor cell, in other cases it's more macrophages or myeloid component, and in some cases it's both. And expression on either tumor cells or myeloid cells can inhibit. In the case of myeloid cells, it's probably representation of tumor antigens in the context of PD-L1 that's inhibiting. But the major mechanism seems to be this mechanism that we call adaptive resistance. So the idea is that the tumor cell is not constitutively expressing. This is not like an oncogene-driven expression, but rather it's the tumor sensing that it is under threat by tumor-specific T cells. So how does a tumor cell sense that it is under threat? It is through cytokines, in particular gamma interferon. So an activated Th1 cell or a cytotoxic T cell that's ready to attack is going to be producing gamma interferon. So unless there's a mutation in the gamma interferon signaling pathway in the tumor cell, which you do see in a few % of cases, PD-L1 expression is highly gamma interferon sensitive. So PD-L1 expression can be almost like taken as a marker for an extant anti-tumor immune response living within the patient. And that's very important because what it says is that a lot of patients really do have within their immune system the capacity to recognize their tumor. With sensitive assays, we think it's going to be at least 80% of patients, far more than we get responses in by just blocking, for example, the PD-1 pathway. And already we know this, if you block PD-1 and CTLA-4, you increase the number of, the response rate. And again, this is just scratching the surface.
Can I ask a question about that? Could it be that there are certain attacks on the tumor cell by T cells, which are not caused by gamma interferon, but which are not
Associated.
signaled by, which are not associated with gamma interferon, and therefore are not setting off the PD-L1 response, but nevertheless may indicate that you might be able to use the T cells to attack. So are you missing, could it be that in some of the 80% you're just missing the, what is signaling to you the right, the fact that some T cells might be mobilized?
Yeah, so there's a lot to that. There's so there are clearly far more cytokines and other signals involved in a.
But if you don't see PD-L1 expression, does that mean, oh, your immune system is not, your T cells are not responding well, or that they're just not responding along that pathway?
So they're it means, my feeling is that it means one of two things. Either you missed it on your biopsy, because again, this is not like a BRAF mutation, which is in every tumor cell. It's localized. So you'll see patches of CD8 cells in one area, and that's where you'll see your PD-L1. So you can miss it with your biopsy. Or there are cells, but they're being blocked from getting into the tumor. This is a big area of interest, which are these tumors that seem to be surrounded by macrophages, and you see T cells out in the stroma, but they literally appear to be blocked from getting in. Or there just aren't, without something like a vaccine, there aren't enough T cells that are activated enough. They're in a state of anergy, so this, these various form levels of tolerance that are reversible with more than just blocking PD-1.
But you think if the T cells have managed to get into the tumor, you should be seeing this PD-L1 expression.
If they've gotten in, and if there are enough cells with specificity for tumor antigens.
Okay.
So it's the repertoire is very, very important in this. It's not just the number of cells. And again, there are plenty of tumors where you see lots of cells in there, and you actually don't see PD-L1. People forget about that, but in very early study that Janice Taub, one of our lead pathologists, did 27% of T-cell infiltrated melanomas did not express PD-L1. I mean, there's a lot of variables.
That's really
So
what I'm
So there's no other, it couldn't be that there is some XYZ blockade which we don't even know about yet.
Oh, there's lots of XYZ blockade. There's lots of, I mean, there's lots of X's, Y's, Z's, and everything else.
Double Z, triple Z.
Right, double Z, triple Z. That's why we have over 1,000 clinical trials. Now, I don't know how many of those make sense, but
No,
yeah.
but my question is, could it be that I just don't have PD-1 blockade, but I have a different one which...
Oh, yeah, and that's what they're doing.
Yeah, so virtually all of the combination trials involve a PD-1 pathway blocker. So blockade of the PD-1 pathway right now is foundational. I think there's some scientific sense to that, but the reality is that's really the wave now. Is that because the idea is that if you get the right kind of response, it probably will involve gamma interferon being produced, upregulate PD-L1, an activated T cell will express PD-1. So that pathway will be upregulated, and so the notion is, you know, you might as well always block that pathway.
Just a follow-up, in fact, the opposite view of the previous question is, have you seen in your clinical trials patients who have high PD-L1 expression, have large mutational burden, high density of CD8 T cells, and yet do not respond?
Absolutely, all the time.
What proportion of this?
I would say it depends on the tumor, but for a lot of cancers, if you have a good PD-L1, well, I mean, let's just take lung cancer in the Merck case. I mean, the response rate in patients who are PD-L1 over 50% on the tumor was about 50%. So there's an example. So only half of those, now, 50% response rate in metastatic lung cancer is amazing, but that means 50% of PD-L1 high patients did not respond to an outstanding PD-1 pathway blocker.
But to also put another layer in, the data that was presented at AACR from the nivolumab of the first-line lung cancer trial, which looked at both PD-L1 and mutational burden, showed that if patients had both a high PD-L1 and the highest tercile mutational burden, they had 70 to 80%. Actually, Alex was the chair of that session. She probably knows the data better than I, but it was closer to 70 or 80%.
Right, but what was the mutational load cutoff for that?
Highest tercile.
Right. So, yeah, so you can, and I'll show you a couple of
So
slides.
they're modifiers, right?
Yes, there
You
are,
showed
there,
that the mutational burden in that case had a dominant effect over the PD-L1, but there's a lot of complexity to these different factors.
yes, which is why, again, there's a lot of complexity. If they don't all overlap. I mean, just looking at this again, I mean, one of the points of this slide is that there is far from perfect overlap. And so, and as we, again, now can start to measure T cell repertoire, the function of these T cells, we are clearly generating more and more variables that biology tells us is relevant. And being able to figure out how to put those together to get the best predictive value is something that we really need to do. And that's also going to give us information that's going to guide us on what additional therapeutic interventions we need to add to that.
So, in terms of thinking about,
All right, first.
okay, in terms of thinking about data on which we might apply some machine learning, some of these, like PD-L1 expression, are probably really not very high dimensional, probably one dimensional or something, probably a single parameter there. However, for mutational burden I had asked you about that, but also for T-cell repertoire and some of these other ones, these are actually much higher dimensional than this thing that you're saying you
Yes,
do
yes.
have a height. And your T-cell repertoire, I mean, I know that Harlan has one number which indicates how good your immune response is, but of course, it's a multidimensional thing.
Right, so-
So there is a much higher dimensional latent variable
Well, the
piece
question
here,
is,
right?
where do the other principal components come from? I mean, if you're explaining some segment of the variance with T-cell inflamed microenvironment or PD-L1, some component with mutational burden, what other principal components do we need to identify to explain enough of the variance? that
So
when we-
T-cell repertoire and
Yeah.
function, and again, there, I am sure we're gonna find patients with great T-cell repertoires, but very few T cells in the tumor because those tumors have developed a mechanism to keep T cells
No,
out.
but I'm saying
But-
to call T-cell repertoire kind of a one-dimensional thing. or
It's
mutational
as you'll
a
see.
one-dimensional thing.
You'll see,
What we
there are
would
many,
love
it's
is a
multidimensional.
high-dimensional
The thing I was getting to
space,
was,
yeah.
right, looking at the node biomarkers may be a low dimensionality problem, but finding those additional principal components that explain variance is a high dimensionality problem, because there's a lot of places to
And
look
that's,
for that.
right, and that's what we, so some of these you actually have more data than you're using,
All of them.
right? Right, and that for us is very exciting, because that is where it may be that there are combinations, you know, that you wanna go out to this very high dimensional space and then try to figure out the relevant subspaces for response to various treatments.
Your challenge would be, how do you create a training set with the relevant number of features that captures this knowledge? That's the
But,
challenge.
you know, sometimes you don't need to have it in every, I mean, there are ways of saying, I don't have to have every patient showing up with all of these things to be able to do it. You can transfer across them, so you can have sparse data sets. It's not like it has to be that every patient has to, you know, we have to know this. There are ways of transferring things across sparser data sets.
I think
So-
there's a question here of, you basically have some large pillars of data that you're working with. For mutational burden, you have sequencing data, from those who are familiar with that, basically you're talking about raw reads from PD-L1 expression, you're talking really about looking at an image and assessing a system per cell. And similar for CD8 density. So all of this comes down, and even for T-cell repertoire, you have sequencing. So all of this comes down to a feature extraction step. So you're
Exactly.
extracting mutational burden as one feature, then potentially you're extracting neoantigen burden as
Right,
another
but I would
feature.
like to go to look much more carefully and say the features I would like to try to extract are before you collapse it all down, because it may be that those, or the combinations of those
Sure.
are more
And
predictive.
some of these don't
right?
even have very well-defined features to be extracted from, as you alluded to the fact that T-cell repertoires we're beginning to come up with interesting indices to come out of that data. So some of these are much more refined in terms of the features we want to extract, like PD-L1 expression, and maybe an intermediate degree of maturation in how we extract features with mutational burden and
Right.
subsequent things. And then with T-cell repertoire, we're still,
Right, but like Harlan's special sauce, some of it is reducing all these measurements he's doing to one number, right? And we might want more of that before he reduces it to a single metric that's interpretable by, by your average oncologist in Kalamazoo, right? I don't know. Do you know what I'm saying?
All right, well, it's all Jennifer's fault. if I-
Once you get to what you really.
Okay. Yeah, Mark wants to see. Okay, so we're gonna, so this is a new assay for T-cell, what, question.
But even if we had the first biomarker, how is that helping us to raise the plateau level? I mean, we're just able to predict where which are the patients who survive, but are we raising also the plateau level?
Yeah, so to raise the plateau level, so first, it is, we also believe that these biomarkers are gonna give us key information as to who's gonna need additional immunotherapies, and I call any therapy an immunotherapy because there's no cancer treatment that doesn't affect the immune system. And we might as well leverage it in our favor instead of shooting it in the foot. And also, it will guide us as to which of this now expanding arsenal of immunotherapies should we actually choose for Mr. Smith and Mrs. Jones. So it is going to start by telling us with more precision who will respond to a PD-1 pathway blocker. A number of these, though, are going to be near-term post-treatment initiation biomarkers that will say, okay, Mrs. Jones doesn't need anything more. Just leave her on anti-PD-1. Mr. Smith, he's going to need something more. He's going to need an anti-LAG3. He's going to need an anti-CTLA-4. He's going to need, a HER vaccine, et cetera, et cetera. So absolutely, as this evolves, this is going to raise the plateau. I think at this year's ASCO, we're going to start to first really see some of the combinations of the many that are beyond CTLA-4 and PD-1 that are beginning to raise the bar.
We should let
Let's go on.
Okay.
Now we've reached the saturation
Okay.
point. about-
Okay, so, all right, so I'm not gonna take any questions till the end. All right, so Manifest really came out of the frustrations that we had at kind of the standard test that people have been using for, 20, 30 years to measure T-cell responses in the peripheral blood, which is the ELISpot, which is fine for viruses where you get very big responses against a small number of epitopes. But anybody who's been doing ELISpots in the cancer world will tell you that it pretty much stinks. The other way is tetramers, and tetramers are great if you know the antigen, if you want to focus in on repertoires and responses to a few antigens. But if you want to look at these broad repertoire analyses in cancer patients, you can't do that with tetramers. People have, so I think one of the real revolutionary technologies, the one that Harlan Robbins developed, is TCR-seq. He likes to call it Immuno-seq because that's adaptive term. We call it TCR-seq, but it basically does deep sequencing of the CDR3 regions to give you analysis of clonal expansion. So what this is essentially integrating, the problem with essentially all the TCR-seq that you've ever seen in the now hundreds of papers that use it is that the TCR sequences are never linked to antigen recognition. So you have no clue what antigens are being recognized when you see an expanded clone of T cells. So Manifest really is about integrating those two. Kelly has turned into a pretty simple. As you'll see, it's sensitive, it's quite sensitive, specific, and it's information rich. And this is where now this whole, how do we actually handle this information? So Manifest stands for mutation-associated neoantigen. We call those MANAs, functional expansion of T cells. But it can be also used if you want to look at antiviral responses. We're actually using this now in earnest to look at endogenous responses to these human endogenous retroviruses that are upregulated in a lot of cancers, very interesting potential shared antigen, or these tumor-associated shared antigens. So we call it ViraFest, TAA-Fest, and if you're drinking beer while you're doing it, it's called Oktoberfest. So this is basically how it works for Manifest. So you do whole exome sequencing, you run it through NetMHC scan, the various prediction algorithms, and as we've discussed, these prediction algorithms are highly, highly imperfect. They're based largely on predicted binding to the HLA alleles in that patient, those predictions are highly imperfect. Some of these new versions try to take into account proteasomal cleavage sites, tap transporter preferred sequences, ERAP trimming. There are many, many steps that actually determine what gets efficiently, which peptides get efficiently presented on MHC on the surface of the cell. And there are in that process a lot of known unknowns and unknown unknowns. And so the list that you get is, which you can go all the way down, maybe 50, maybe 80. But if you now were to look at T cell responsiveness, the top 3 responses would not be against peptides 1, 2, and 3 on the list. It might be 7, it might be 16, and sometimes you even go below the cutoff that they tell you can't respond below that. So what this does is so you generate your list, synthesize the peptides. The nice thing is that you don't actually have to use purified peptides. The crude peptides actually work just as well, which from a cost standpoint is a big deal. You do a rapid T cell purification. Again, Kelly's made this very turnkey. You basically throw in your peptides. It's a 10-day culture. We know that with this culture, you don't expand naive T cells. So if you look at, if you look at Ebola antigens or Zika antigens or HIV antigens from donors that have not been infected with these viruses, the epitopes have been defined. You don't see that you're pulling them out with your standard 10-day culture. If you want to pull out naive repertoire, you can do a second repeat stimulation, and then you actually do pull them out. The key is that the readout now is not cytokine production, but it's essentially TCR-seq, basically around the CDR3 region, which pretty much gives you the diversity. We generally do just TCR-beta, but one can begin to now do the so-called pair-seq, where you incorporate your alpha. But as you'll see, there's a lot of information from TCR-beta. So let me just give you one example. So one of the kinds of patients we're very interested in are the patients with low mutational burden, and I'll show you an example of that do respond. But Jennifer was asking about the dimensionality of the readout. So here, just thinking top view, 5 different dimensions. There are the number of manas that are recognized. There are the number of T cell clones that are expanded to a given mana, or in toto, if you add up all the positive manas times all of the clones that are expanded per mana. There is the magnitude of the expansion. We think this will actually give us information about the activity state of the T cell, because we know that an anergic T cell will proliferate, but it'll only give you maybe 2 rounds of proliferation, and then it sort of peters out, whereas a good effector memory cell can give you 10 or more rounds of proliferation. So there's the magnitude of expansion. You can then take these clones, these CDR3s that expand in this in vitro manifest assay, and again, what you're looking at is post versus pre-expansion. You're actually looking at no peptide control as your level of expansion. So there is the magnitude of the expansion. Then you can also go back to the biopsy of the original tumor, the archival biopsy, do your TCR-seq on that, and look at the representation of clones expanded in the manifest assay that were actually in the original tumor biopsy, and I'll show you an example of that. And then there's this whole thing, and JW, who's an MD PhD student, who makes me very glad that I'm not applying to the MD PhD program at Hopkins now, because I'd never have a shot, has this immunomap algorithm that he's been working on with Alex, that, let's say, looks at the quality of the mana-specific repertoire. And this I'll be very interested to get. So here's this patient with a complete responder, lovely lady, with only 30 mutations in her lung cancer. We've actually, together with Memorial, have been collecting. We have about 12 or 15 lung cancer patients between the two institutions with less than 50 mutations, so low mutational burden, that have had really great responses to various of the anti-PD-1 antibodies. So if you run the NET-MHC scan, it tells you there are only 12 predicted neoantigens. So Kelly actually made 80 peptides, so she just went down the list. So basically 68 peptides that the algorithm said would not a snowball's chance in hell of being recognized. So the first thing she did was an ELISpot analysis, and she found one peptide that was ELISpot positive, which was peptide 25. So already it's way below the prediction, which is on the order of the typical is predicted 500. I think it's 500 nanomolar is the prediction. So you can see it's about twofold above background, but it was consistently positive. But these are the kinds of things you see with ELISpot. So with Manifest, she found of these 80 peptides, 29 peptides in which there was a positive response to. So what does that look like? Well, if you take your peptide 25, you get a lot of clones that are expanded. So these are all the positive peptides here. This is read out like a heat map, so red is highly statistically significantly expanded. And remember, for a given CDR3 that expands to peptide 25, the expansion of that CDR3 to all of the other peptides essentially becomes the negative control. So you've got a lot of negative controls, and that's what gives you your statistical power. So a couple of these, interestingly, you do see in others, but most of these you see basically flatline for all of the rest of the peptides. So you start to look at p-values, and the program spits out p-values of 10 to the minus 212. I mean, the statistical power is really amazing here. So here's an example of one peptide that was negative by ELISpot. There are fewer clones, so generally when you do see an ELISpot positive, you do see a gazillion expanded clones in the manifest. If it's ELISpot negative, you see something like this, still plenty. And again, the statistical power is just really huge. So this is all of the positive peptides compressed. And you can see that these reds here, if you go across, it's only this peptide. This is peptide 25. They're clustered according to their sequence similarity. So you can see peptide 25, there are a number of these clusters that are positive. So this has brought up for us the notion that the anti-tumor response that you bring up with anti-PD-1, again, this is a responding patient, may look more like an allo response than a viral response. In other words, it's the sum of many small responses rather than a few really big responses. I have to say, this is exactly the opposite of what Steve Rosenberg will tell you, which is that there are only 2 or 3 peptides that you ever get a response to. But of course, it's all according to the sensitivity of the assay. Now, again, if you take peptide 25 and you say, okay, which of those, 150 CDR3s that expanded in vitro, can you actually go back and find that exact CDR3 in the tumor? You can actually find 4 that were actually present in the tumor. So this is yet another variable that may tell you which of these peptides among the 29 might be the most relevant by looking at the tumor. But then there's this whole area of sort of the richness of the TCR repertoire that expands in response to a given peptide. And this is where JW comes in with this immunomap analysis. And essentially, it's an evolutionary tree algorithm. And please do not ask me any questions on this because I'm just a dumb immunologist. That essentially looks at TCR relatedness. So this is what, now remember, this is not divergent evolution, this is convergent evolution because the TCR primary repertoire is generated at random, and so this is what's converging in response to this given peptide. And so these lines and distances essentially represent sequence distance. And what you can begin to see is that there are clusters. So you can come up with a diversity score, which basically tells you how many clusters you have. You can then look within a cluster at how many CDR3s are within a cluster, and JW calls that richness. So you've got diversity, you've got richness, and again, this is all generating, essentially algorithms to come out with a metric, and he'll be the first to tell you that there may be better ways to do that, and he just loves to get into this stuff. And then you can also look at expansion of single clones, which you can represent essentially by the size of a circle around a single point, which is a single clone. So what does this tell you? We're learning, but here's just an interesting example. So these are 2 patients. This is the patient that I showed you with 30 mutations, 29 positive peptides recognized on manifest. So each one of these dots is one of those peptides. So if you add this up, there are 29 dots here. This is another patient that also had a number of positive manifest peptides. I think this patient had about 20 or so, who's a non-responder. And now this is looking at the motif analysis, which is the number of clusters, essentially the richness of the motifs, and a diversity score. These are all generated by a set of equations. And what you see is a couple things. Firstly, particularly with the motif analysis and the richness analysis, the responding patient is highly statistically significantly higher when you look at their clones than the non-responding patient. We obviously we need to do 100, 1,000 patients. The other thing that's really cool is that there were a couple peptides that stood out as having much higher numbers of motifs. So this is actually, turns out to be peptide 25. These are some of the others. These 4 are basically these 4. The diversity score, less informative, at least comparing these 2 patients. And what these reds are going back and looking at CDR3s in the archival biopsy, so in the TIL, that cluster with the CDR3s that came out of the manifest assay. And there were 5 peptides, 4 of which were these guys that stood out on motif analysis and richness analysis, that cluster with sequences seen actually in the tumor biopsy that add up to greater than 1% of the total clone. It's not really clones, it's really clusters within the tumor. Now again, Do we know that these clusters are representative of T cells that see this peptide? No. For that, we actually need to do tetramer sorting and look at that. The point I simply wanted to make here is that we think that these may be a set of analyses that can begin to distinguish, because this non-responder was also a patient with roughly 40 or 50 mutations in their tumor. And so, and actually, I don't think there was a huge difference that was obvious in the biopsies in PD-L1 expression. So this is an example where this repertoire analysis may give us additional useful information that may be predictive.
Hey, Drew, can I ask a question? I know you wanted the whole questions, but I'm wondering if you're measuring more than one thing here. Because what you were showing earlier is that the frequency of some of these responding clones is actually higher in the peripheral blood than in the Till compartment. Some of these are clearly highly represented in the Till compartment.
That's after expansion. So you're looking at that.
So that's a post-expansion.
Yeah, so this is not post-expansion, because this is what's in the tumor. This is post-expansion. In fact, yes, this is
So
post-expansion.
you can't really do a quantitative comparison.
No, in fact, if you go back and look in the peripheral blood at these, that's the equivalent of no
But you don't
peptide.
find all
You
the
actually
clones,
don't.
but you don't find all the clones that you find in the periphery in the tumor, right?
That is correct. The caveat being, we're looking at a little sliver of biopsy that we send off.
So my question is, in terms of measuring more than one thing, I wonder if some of what you're doing with the peripheral blood is actually priming naive T cells that have, the potential reactivity here. And so you're actually, in part, measuring systemic immune competence to actually get de novo priming. And that's being mixed together with the detection of some neoepitope-specific T cells that are actually in the tumor.
Right, so that's a good point. So the one thing I can tell you again is that Kelly looked at least at the question of if you take peptides that are known peptides from viruses that can be recognized, like HIV, Zika, and whatnot, and look at and apply manifest to those peptides in individual donors who we know were not exposed to these viruses, with the 10-day culture analysis that all of these use, you don't ever see expansion. If you do a 10-day re-stimulation, you do, which I think is very cool, because I think you can titrate your culture conditions to, you sort of in a sliding scale to look at what is more available in the repertoire to expand relatively quickly versus what's potential that you need to actually push a lot harder to be able to see. So again, that's arguably yet another parameter. And this is one of the sort of embarrassment of Rich's problems that we have here, is we're generating a lot of data, and we now want to understand which of those matter. So the first inkling that that mutational load might actually matter for responsiveness is that the high mutation burden cancer types tend to be the ones that respond better, melanoma and lung cancer. The best example of that are these genetic subsets of cancers that are microsatellite unstable, that are mismatch repair defective. Half of these cancers are in patients that come from Lynch syndrome families, hereditary non-polyposis coli. And basically, this was when in a brainstorming session in 2012 came the notion that was really when cancer genetics and cancer immunology got married. So the hypothesis was that, that if you are MSI or mismatch repair deficient tumor, you've got typically more than 1,000 mutations. You should be as antigenic as hell, so you should really respond well to anti-PD-1. And this was kind of one of those clinical trials that you sort of dream of. So red are mismatch repair proficient colon cancers is a waterfall plot. Green are mismatch repair deficient colon cancers, and the black are mismatch repair deficient non-colorectal cancers, of which, endometrial cancer, it's about a quarter, 20% of gastric, 3% of prostate, et cetera, et cetera. So they got married. I just have to show this slide because it shows you there are real people here. So that's the history of the definition of the genes involved in Lynch syndrome. Unbeknownst to the cancer genetics people, PD-1 was being discovered, defined as a checkpoint. The first paper on PD-1 blockade was in 2010. The clinical trial, Merck was kind enough to give us pembrolizumab, but you wouldn't pay for the trial. But Bert paid for the trial because he's got more money than God. And about that time, a 22-year-old named Stephanie Joho, who turns out to be from a Lynch family, was diagnosed with colon cancer metastatic in 2014, sent home basically to die, 100 pounds, entered the trial in 2014, and that's her today. She's 14 months in CR and just celebrated her 27th birthday. So this is real people that science actually makes a difference in. So this is a little snippet of her immune system. Here are 3 manas in which there are T-cell clones expanded that we could find in her archival tumor. These 2 were from frame shifts. This was a missense mutation. The wild type version of this peptide has about 3 logs lower affinity for the restricting HLA allele. And this is what happens to these clones in her blood. Basically, you can see that at about a week after initiation of PD-1 therapy, of these 2 clones that see this particular peptide, one never goes up in the blood, but look how fast this shoots up, and then it comes down. It looks almost like a viral response. These 3 clones, this one shoots up very quickly. This one takes 14 days to peak. This one takes 22 days to peak. These 2 particular clones against this peptide, one of them never increases. The other shoots up at the first week after initiation of PD-1 therapy. So this is my last slide. So again, just to introduce that, now we've got some really interesting new assays. We're very excited about this. We've been talking with Harlan about actually separating out a unit in which people can actually send just frozen peripheral blood cells to, so that there can be real consistency in the in vitro culture part of the assay. Then you do your TCR-seq, and then you've got basically data that you can play with in terms of algorithms. So
I'm going to try and be really brief, try and catch up a little bit. I'm going to try and really be brief, focused on what I want to tell you guys a little bit about, and maybe we can follow up later and try and save some time. So I work at Johns Hopkins as a pathologist and as an informatics person there. And when my primary background was in the genomics space, and then obviously ventured into the immuno-oncology, immunogenomics space as well. And when I first started talking to Drew, immediately kind of realized that there's a lot of great data there, and the first step, I think, towards analyzing it is organizing it. You can't organize it all together into a framework, and then you can keep ingesting data into it. It's not scalable or sustainable, and I think the TCGA addressed that same scenario when they started realizing that they have noodles of sequencing data reaming out. They got to organize it some way and present it to the public. So just as a show of hands, who's familiar with, first is base. I'm sure everybody's familiar with TCGA, I assume. I'm primarily asking like Microsoft folks, just to understand what kind of context here.
Just a little
Genome
bit less
Atlas?
now.
Okay, good.
So yeah, the TCGA was like a huge sequencing effort over the last 10 years, kind of, in some sense, revolutionized some of our understanding of tumor genomics and some of the transcriptomics, the expression of genes. That's like a fully publicly available data set, which then has had 2 pieces of software, open source, developed to help organize and enable the public and the data scientists to engage that data. One is the Genomics Data Commons, okay, which is a joint effort primarily between the Chicago group and the NCI, along with some other places as well. A lot of people have contributed to it. It's primarily an archival system to help you identify what data you have, can I get it out, and then analyze it. But at least it's organized in one framework. And then the other open source piece of software that was built around the TCGA was the CBio portal. That's more of like an analytic framework on the top. I would say it's analysis light. It's not like for the data scientists, but it just gives people, the non-data scientists, the ability to engage and ask questions of the data. So when you hear a person like Drew talk about this data, it's very complex. And certainly, again, a person with domain expertise in immunology can ask very pointed questions of the data. So there's 2 basically views of data, right? One is that you're going to take a domain expert, they're going to have a pointed view of what they want to ask, and you want to have to enable an interface for them like this. And the other view that you also have to account for is a much more global view of the data that you can apply more general pattern recognition tools or other statistical methods to identify patterns that may not have been apparent to the domain expert. But those come with different trade-offs. As many of the data science people know, they have different trade-offs. Neither one is perfect. So I'm not going to talk about the background of immuno-oncology. I think it's been summarized quite well. But what I will highlight is that, as was shown by some of the survival curves that Drew showed, what we really need to understand is how are we going to tackle developing biomarkers, characterizing them as predictive and or prognostic, and being able to actually make that distinction, which is, I think, really important and sometimes overlooked. So I'll stop there. Everybody's familiar with the distinction between the biomarker that's predictive versus prognostic, and why that could be a huge, it's be a big confounder if you tried to take a prognostic biomarker and claim that it was a predictive to response if you didn't have the appropriate study design set up. So, yes?
Yeah, I would say clarify that.
Clarify. Okay.
A lot of oncologists don't even know.
So a prognostic marker is one that effectively says, if I have a tumor and I sample it and I don't do anything and I just observe the natural history of the disease, this feature will tell me who will do better or worse, effectively focusing on the natural course of disease.
It certainly came from the days that we didn't have.
I agree.
a lot to offer.
Right, and so that's why it becomes very difficult to think about these types of questions when it's hard to really understand sometimes what the natural history of certain diseases are if most of the data you have about them is seeing some form of treatment. But nonetheless, you have to at least try, and you have to be at least cognizant of it. And then a purely predictive biomarker is one that doesn't really have anything to do with this natural history of what's going to happen to a patient of a given feature, but it actually predicts response to a drug. Right, that's what I said. That's why I said then there's biomarkers that are predictive and or prognostic, and but it's important that we think about that, I think. And in order to get to that point, we have to have large, well-curated databases harnessing much of the high-throughput data that some of which Drew has alluded to from the genomic side, from sequencing in particular, and various interesting assays that leverage next-generation high-throughput sequencing. And then also pairing them to outcomes for patients in terms of, again, what drugs they received, how'd they do, and normalizing all that into one framework. Yeah?
When you say large, I wish large is possible, but what large is practical?
Practical. Okay, well, the TCGA is practical, and it holds 10 to 15K. The efforts that we've done through the American Association of Cancer Research through our GENIE consortium covering clinical sequencing is 20K growing to 40. We know that the immuno-oncology trial space is somewhere to the tune of 100 to 300K patients. So that would be a great target to aim for. But I think one of the caveats with that, and I would, anybody who's in industry, I'd like to here how much data sharing can happen with trial data. You know, I mean, that's like, yeah, it's a problem. We face that problem in that consortium I mentioned, that clinical sequencing consortium, because we really are only able to easily share the data for patients that are not on some type of trial where there's a sponsor covering it, right? We have an obligation, at least at Hopkins, that's at least the contracts that we have. So there are significant regulatory and contractual considerations when trying to get at the key data we really want. But over time, in scenarios where drugs are no longer on trial, like for example in melanoma, if that's going to be a front line, or in lung where it's going to be towards front line as well, those settings you can obviously look at that data. It's not a trial anymore. It's standard of care. So that's the game we're going to be playing here, I think, is trying to have the data sets accessible and, you know. So.
You retroactively get the data from the trial, or is that protected by?
I do not know the answer to that question. I'm sorry. From my understanding, it depends, but I'm a little too junior to be sure of that, because I think anything can be negotiated, I think, but it depends. So I put the data into 3 spheres. There's immunogenomics, there's immunophenotyping, and immuno-oncology. So much of what we were talking about previously has to do with immunogenomics. So the application of next-generation high-throughput sequencing to characterize the tumor's immunogenicity along with the immune system's repertoire to detect those things that in a sense, the tumor's throwing up there for the immune system to sample. There's immunophenotyping, which characterizes the type of immune cells engaging the tumor along with their quality. And then there's immuno-oncology, which speaks to the clinical covariates that are really relevant to the IO space, which is different than what was relevant in conventional targeted or chemotherapeutic, conventional chemotherapeutic approaches. And I highlighted the difference between predictive and prognostic. What you see on the right, and I'm going to dive into a few corners of it, is the middle portion here. I mentioned the genomics data commons, right? So it would be silly to reinvent wheels, especially if they're open source and available. So the centerpiece here is the main data model for the genomics data commons, linking primarily NGS, next generation sequencing data at the genetic and transcriptomic level with clinical outcomes. So I invite all you guys should go take a look at that website, and the GitHub site is available. You can copy it, set it up yourself. The underlying structure is a Postgres database with an object relational mapper on top of it using SQL alchemy to convert it to a different type of model here. Rather than conventional tables and Postgres, it's more of a graph model, which enables a little bit of flexibility in how we're going to talk about different data elements. So jumping right in, one piece is immunogenomics. So we would start with, these are very small, but the point of this is to show that the starting point is actually reads. So let me just take one pause and say, when I say a read from a sequencing run, everybody knows kind of what I'm talking about. Basically, the genome is basically a string, a polymer, and it has a defined alphabet. So when we do sequencing, right now most of the technology, there's others that can do longer reads. Most of them are in the tune of hundreds of base pairs. And then we just get millions of reads like that. And then we have to figure out what we want to do with that. That's the raw data we're starting with. So
are you just doing the genome, are you doing the exome, are you doing?
As you can see, there's a variety of different approaches we can do to sampling. The genome is the entirety. Yeah, the genome would be the entirety of the sequencing effort. And then there's further subsets of the genome that can be analyzed, obviously. Genome, exome, small, non-coding, limited panels, whatever. We would want to do exomes, because exome has a clear implication towards neoantigen, right? Because unless some major things change, there's going to be a peptide in that MHC, presenter. So you're talking about things that are protein. So proteins are encoded, generally speaking, from genes. So you're talking about the coding sequence.
But you're going to miss the regulation aspect of it a little bit.
Right, and so that's why people have. But then you also want to consider looking at the transcriptome, what is actually expressed into RNA, and that can further give you more information. And then what I'm showing on the right is the additional data model that we would like to overlay on top of the genomics data commons, which is really taking into account mutational burden, neoantigen prediction from mutational burden, along with potentially RNA level data, functionally validating these. And then another arm of it says, what if we start with TCR-seq data, which Drew already described, characterize the diversity, characterize the phylogeny that's in that data, and then link the two together to be able to identify potentially that's what Drew's trying to show, is a link between what are the TCR repertoire that's out there and what peptides are potentially, how do we make that connection? Because if you could make that connection, then there's a lot of implications in terms of how you would potentially consider targeting therapy for a patient. For immunophenotype, what's that? Yes, those are also genomics approaches as well that you can do these days, sequencing at a single cell level. And the reason that's really valuable is because you have some limitation in your sampling when you do if you just take a piece of tissue and grind it up and do sequencing, if you're looking for a needle in a haystack, and sometimes we're looking for characterizing which tumor, like PD-L1 positivity in some tumor types is down to like percentage points. So sometimes it might be hard to pick up that difference if you're looking at the whole homogenate. But if you had single-cell approaches, and some of these single-cell approaches apply not only to genomic characterizations, but expression characterizations as well. And that's really an evolving aspect of this characterization, moving potentially away from grind up the whole tissue and analyze it just as an en masse and really be able to do a single-cell based. So instead of having one data set, it just adds more complexity, right? So instead of your data matrix being, you have samples and you have tons of variables, now you take that one vector that used to be your sample vector, and that becomes two-dimensional as well, because you can look at all the different events that you're characterizing. And how to approach those types of analyses. There are obviously some great techniques that have already been developed to address that, particularly from the flow cytometry world, where data comes out that way in a single-cell nature to begin with. But these are the kind of things forward thinking in terms of where immunogenomics probably wants to go. Immunophenotyping, I mentioned earlier, refers to a variety of things, but for this discussion, one of the main things we'd want to be able to think about is when you do take a tumor and you do RNA expression on a grind up, just grind up the whole mass of the tumor, you're admixing quite a few cell types, including epithelial and immune and stroma and other, vascular. So what if you could tease out from that, what if you could deconvolute from that signal, if you will, that came out, that vector that comes out of one sample, you could deconvolute out different immune signatures. What is the activated T signature that's potentially present in there? So there are active areas of research that people are developing immune signatures that are trying to tease out if there is an immune component in there, what is it potentially trying to tell us about its engagement with the tumor? And then as Drew alluded to also being able to do this, yeah?
So does it use other data to decide that, or is it just some...
You are the only participant in the conference. Please use your keypad to remain in the conference.
Or is it just some mixed membership model representation?
There's a variety. So this question of deconvoluting out gene expression signatures has been around for now more than 10 years. So there are a variety of different approaches. Some need outside data to deconvolute out the signal. Some do it by themselves. There's a spectrum of them. And there's even now they have a whole database of quote unquote signatures that are out there. You can there's a molecular signatures database that spans immunologic signatures, epithelial signatures, all of which should be something relevant to incorporate in a large data commons. And again, as Drew alluded to, there's an important component here of understanding the spatial relationships that between the immune cells and the tumor, which we can see on the slide as well as a pathologist. I mean, as a pathologist, I joke around. I've been staring at immune cells for my professional life. Most of the time,
You are alone in the conference.
I am alone. I am alone. So all of this really comes down to, I think, trying to characterize this very exquisitely complex immune synapse, right? The engagement of this T cell looking at antigen-presenting cells, okay, or tumor, and trying to understand first of all its components, understand where can it be modulated, and really having a respect for how I mean, it's just really complex what's actually happening. Many of the stick diagrams we showed talk about 2 different molecules pinging together and good. It's actually very complicated, including tons of molecules, double A through quadruple Z, whatever, which at the center point we think about being the peptide MHC with the receptor. And then how the T cell actually physically goes in, hits the target, spreads out, and then how the sensors, even on this surface, change in their physical position. It's amazingly complex. So it's not.
Do you get some in the spatial data you were just talking about? Can you capture this kind of thing? Or
Not on my microscopes.
no, but I'm saying would you have like RNA-seq data or something that would reflect the expression here, and you'd see the T
In
cell,
my
you'd
domain
see the
expertise,
tumor.
yeah, in my domain expertise as a pathologist, my resolution usually goes down to like, 5, 10, microns. I mean, that's where I really can tell stuff real well with like a high-powered light microscope, and I can see the interaction between cells. But with this kind of stuff here, this is a schematic of something that's taken a lot of different assays to actually tease out of what's happening on the surface of the immune synapse. So I. Yeah. Yeah, there are. There are super resolution techniques. There are a whole bunch of different techniques out there, but I'm just saying for clinical practice, what I can look at, we can discuss that in a different. Yeah, we can talk about the imaging stuff, I think, at a later point. My point in this slide was just to highlight that we've been talking about T-cell repertoire, and we've been talking about peptides, and I wanted to give a visual of how much more complex. There's more factors here to take into account. But we can begin at least to characterize some of the components. And I think that's what TCR-seq does. That's what what Manifest is trying to do, is trying to figure out what are we playing with here. And we know some of the other surface receptors that we're modulating with drugs. So this is where the game is being played, is in this interface. And then, of course, I mentioned that there's like key pieces about immuno-oncology. We have to have, which unfortunately, in the clinical trial space, the data is abstracted beautifully, generally speaking. That's our gold standard. So if the clinical trial data sets are not good enough for clinical annotation, then we're in a lot of trouble. In terms of routine clinical care, the data that we really want to target, which is going to be accessible for large-scale data analysis, that's where the problem exists. Because extracting well-curated data from non-clinical trial EMRs, if anybody does that, they know how much, that's like half my resources as my investigations go into that process. Getting those annotations about what happened to patients that are not in the context of a well-defined trial with a form and all that kind of good jazz.
Alex, would you say that systems like Epic are... Well, it's what half the hospitals are
I
using.
know. We don't want to have the.
What I'll say about Epic is...
Epic is a billing system.
The Epic is a billing system, I agree. I will say one thing about... I'll say this about Epic. What were you going to ask first?
So I was going to ask whether current systems are improving clinical annotation in non-clinical trials.
So my answer to that is that I think the jury's still out. I think that the technology actually is in place. In other words, you can do this if you wanted to. The problem, and I hope not to offend any clinicians here, I am a pathologist, but you say the naughty word to fill out a form to a clinician in routine practice, and they're going to stare back at you and say, heck no, right? They're going to say, I've got 40 patients to do today. You're absolutely out of your mind if you're going to add time to my day. So I was talking to some of the Microsoft people earlier that really one of the things I harp on is that what we're facing in the next decade is a data entry user interface problem, in the sense that we need a way that clinicians can do the data entry when they're doing routine care in a manner that's just as fast as them picking up their thing and saying, patient Bob, yeah? Yeah, I'm actually done. Yeah. So the bottom line is that that's going to be a key piece going forward, because right now we have two paths. One is we got to deal with all the retrospective data we have, and we have to harness that somehow. Second is we have to find a way prospectively going forward to not be in the same situation we are now 10 years from now. And all of this, vision I have for what I'd like to do, at least at Hopkins for our data, is to really enable this two-tiered data access. To speak to that is we would put like something like the Portal on top of this data commons so that anybody can go in and make simple univariate associations and look at their favorite analyte and say, I wonder how that analyte looks like in the context of this mutation. What does survival look like? What did outcome look like? But the problem is I have to have outcome. Right? I have to have that data. If I don't have that data, I can't make any associations. And then, of course, underneath, a data scientist can go in with tools like R, Python, whatever, and do all that.